floor is yours.
Okay. Thank you. Just wait for my slides. So it says drug target MR. Yeah. Okay. Yeah. So thank you, Abba. So I'm going to be discussing drug target Mendelian randomization and both some of the strengths and also potential challenges that can arise when we apply this method to dementia research. So as a bit of a background, as I'm sure many of us are aware, dementia and cardiovascular disease appear to share many important cardiometabolic risk factors. And some approved medications that target these risk factors have been linked to reduced risk of dementia in conventional observational studies. So this suggests the exciting prospect of potentially in some way repurposing these medications for dementia prevention. However, as we all know, though we can take various steps to try to minimize sources of confounding and bias in conventional observational studies, these frequently persist. And thus the causal nature of associations between medications and dementia risk is often unclear, and thus the efficacy of these medications as repurposing agents remains uncertain. So one alternative approach that we can use to try to estimate effects of medications on dementia risk is drug target Mendelian randomization, which is a special class of Mendelian randomization, which has been discussed previously. In drug target Mendelian randomization, we take one or more germline genetic variants located in or in proximity to genes that encode drug targets of interest, and we use these as instruments or as proxies for these drug targets to try to estimate their effect on disease outcomes like dementia. And the reason we use germline genetic variants as instruments or as proxies is because of various favorable properties governing their inheritance, particularly molecular traits like protein targets of medications. So for example, germline genetic variants, as we know, are those not in LD with each are random meiosis. This should minimize conventional issues of confounding. Germline genetic variants are fixed at conception and cannot really be altered by subsequent disease status. This minimizes issues of reverse causation. And finally, using modern genotyping technologies, germline genotype can be relatively precisely measured, which minimizes measurement error. How does drug target Mendelian randomization work in practice? So as I mentioned before, we typically construct instruments using variants in or within proximity to genes encoding targets of interest. And we call these cis-acting variants. So for example, if we were interested in instrumenting HMG-CoA reductase, this is the drug target of commonly prescribed LDL cholesterol-lowering medication statins. We would select one or more variants located in or within proximity to the HMG-CR gene, which then subsequently encodes HMG-CoA reductase. And we can weigh the effect of these SNPs by levels of HMG-CoA reductase themselves in the blood, for example, if we had that, or potentially downstream biomarkers of HMG-CoA reductase perturbation, like LDL cholesterol levels, or potentially by expression of the HMG-CR gene in various tissues or in blood or whatever tissues are biologically relevant. It's important to note that in drug target Mendelian randomization, as the name suggests, we're not instrumenting medications per se, but rather the target of these medications. So returning to the statins example, if we're interested in exploring the role of statins, we cannot instrument statins really, we instrument HMG-CoA reductase, this protein that is the target of statins. What this means is in these analyses, we can explore potential on-target or target-mediated effects of a medication, but not off-target effects of a compound, for example. Okay, so historically, drug target Mendelian randomization and also related genetic approaches have been helpful in terms of helping us to prioritize and also deprioritize certain targets for pharmacological development. So very well-known examples of this are PCSK9, interleukin-6 receptor blockade for cardiovascular disease, more recently TYK2 inhibition for psoriasis. And drug target Mendelian randomization has increasingly been applied to examining dementia-related outcomes. And I think it's in dementia where drug target Mendelian randomization offers a particular strength, as the use of germline genetic variants and their study across the life course fills in an important gap on the potential effects of long-term drug target perturbation, given the possibilities of long induction periods for many medications and also known long latency periods in dementia. So this could be helpful, for example, where the lack of effect of a particular drug in a clinical trial, where it's unclear if this is due to a limited follow-up in the trial or due to the particular stage of dementia participants. This does introduce a new complication where if you find an effect using drug target MR in dementia, it may then be unclear over what length of time and what period in the life course a drug may need to be given to confer an effect. Okay, so I wanted to just highlight some applied examples of drug target MR analyses in dementia. So there's some preclinical support to suggest that LDL cholesterol levels may have some important role in Alzheimer's pathogenesis. This is supported by conventional, so prospective studies suggesting that circulating LDL levels appear to be associated with an increased risk of Alzheimer's. And likewise, use of statins, again, this is an LDL cholesterol-lowering medication, is associated with lower risk of Alzheimer's compared to non-users. So this is one of the earliest, I don't think the earliest, but one of the earliest drug target MR analyses that evaluated dementia. And this was led by Dylan Williams, who's here with us. And so in this paper, the authors were interested in evaluating the role of LDL cholesterol, HMG-CoA reductase, in addition to some other lipid-lowering targets, which I won't be focusing on here. So they constructed an instrument for LDL cholesterol using 55 SNPs, strongly and independently associated with LDL cholesterol. They then constructed an instrument for HMG-CoA reductase using variants in or within proximity to, again, the HMG-CR gene strongly associated with LDL cholesterol. The assumption here being that variants in the HMG-CR gene that's strongly associated with LDL cholesterol, this effect is mediated through HMG-CoA reductase. And what they found overall is essentially little or no evidence of effect for either genetic LDL cholesterol or HMG-CoA reductase for Alzheimer's risk. And this is consistent with a large mice trial showing no effect of statin intervention on dementia. And I think this particular MR analysis is important, and it provides some complementary evidence to the clinical trial in that it suggests that the lack of effect of statins in this clinical trial is probably unlikely to be related to the relatively limited follow-up duration, which was around 5 years. Okay, so moving on, I'm sure many of us, sorry. As I'm sure many of us are aware, can you hear me? Yeah. As I'm sure many of us are aware, type 2 diabetes is linked to an increased risk of Alzheimer's. And because of the potential role of insulin resistance and altered glucose metabolism in the brain in Alzheimer's, Alzheimer's has been proposed as type 3 diabetes. Clinical trials of anti-diabetic medications, mainly looking at cognitive functioning outcomes, have been inconsistent. So in this paper published last year in Neurology by Tang et al., the authors were interested in exploring the role of 4 anti-diabetic drug targets in relation to Alzheimer's disease risk. And they found some strong evidence to suggest that genetically proxied inhibition of the target of sulfonylureas appear to be associated with the reduced risk of Alzheimer's. They also found some evidence, though relatively weaker, this should be GLP-1R agonism, was associated with the reduced risk of Alzheimer's as well. So sulfonylureas work by blocking ATP-sensitive potassium channels on the membranes of cells across many tissues. So the activity of this in the pancreas leads to insulin secretion, for example. And KATP is abundantly expressed in the brain. So the authors of this paper, they noted in their paper that sulfonylureas have a poor penetration across the blood-brain barrier. And for this reason, in this paper, the authors expressed caution in the repurposing of sulfonylureas for Alzheimer's. So I just wanted to flag this because clearly, obviously, it is an important issue to consider the blood-brain barrier for a particular drug in a drug target MR analysis of dementia. This is something that's quite unique that we need to think about in dementia. But sulfonylureas not clearly passing through the blood-brain barrier really only represents an obstacle to interpretation if we make the assumption that sulfonylureas have some sort of effect on Alzheimer's through a direct effect in the brain and central nervous system. So clearly, if sulfonylureas after blocking KATP, if this then leads to some kind of downstream effect on circulating molecular traits like glycemic traits, if these subsequently can both pass through the blood-brain barrier and influence Alzheimer's, this is obviously not an issue. So yeah, I just wanted to highlight this because it is a good example of some of the unique issues we need to think about in drug-target Mendelian randomization dementia related to the blood-brain barrier and the need to think about potential alternate mechanisms of effect where a drug of interest clearly does not pass through the blood-brain barrier. Okay, the final example that I wanted to focus on is blood pressure and antihypertensives. So I mean, this is from the Lancet Commission for Dementia Prevention, which was discussed previously, but my understanding is that there's relatively consistent observational evidence to suggest that hypertension, at least in midlife, is associated with an increased risk of dementia. We know from meta-analyses of clinical trials of antihypertensive medications that these appear to lower risk of dementia. So ACE inhibitors are commonly prescribed antihypertensive medications. And in this recent drug target MR analysis, the authors were interested in exploring the role of genetically proxied ACE inhibition on Alzheimer's risk. And what they found, perhaps contrary to what would have been expected, was they found evidence that genetically proxied ACE inhibition appeared to increase risk of Alzheimer's. So there are various ways of, there are a million nuances in this finding, and I'm not going to go into them now, all of them now. But you could think perhaps a possible, though perhaps not very plausible explanation is that perhaps ACE inhibitors do genuinely increase risk of Alzheimer's, perhaps in contrast to all other antihypertensives. Again, that's possible. How plausible that is, I'm not sure. Another possibility is that perhaps lifelong inhibition of ACE, proxied by germline genetic variants, has some sort of differing effect to relatively short-term pharmacological ACE inhibition in mid to late life among individuals with hypertension. Again, that's possible, though perhaps not very plausible. Another possibility here that could at least in part explain these findings is simply survivor bias. So when we think across the life course, individuals with a genetic predisposition to ACE will on average have a lower risk of cardiovascular disease. Cardiovascular disease on average occurs before dementia. What this means is that these individuals will simply be more likely to be alive and perhaps live long enough to develop dementia. So I was interested in exploring this further in this particular analysis. So I used the same instrument, the same genetic instrument the authors used in their paper. I confirmed that genetically proxied ACE inhibition was associated with a lower risk of cardiovascular disease, as you would expect. Again, using the same genetic instrument and also the same Alzheimer's outcome data, I confirmed what they showed in their paper, which is that genetically proxied ACE inhibition appeared to be associated with an increased risk of Alzheimer's. To account for potential survivor bias, I then adjusted this estimate for genetic liability to cardiovascular disease, and I found that this association attenuated toward the null. And indeed, this is similar to some work that was published earlier this year. I think some of the authors are in the room here, where the authors on this paper were interested in systematically evaluating, I think, 10 of the 12 Lancet Commission modifiable risk factors for dementia prevention using Mendelian randomization. Contrary to what I think would have been expected to have been found, they found evidence that smoking, obesity, and blood pressure all appear to be protective against Alzheimer's. I also wanted to note that these findings are a little different to the ones presented by Neil, and maybe we could discuss that later why that might be. Could simply reflect differences in instrumentation or outcome data. So Like me, for the ACE inhibitor example, the authors were wondering whether this could be driven through some sort of survivor bias mechanism. So they adjusted their estimates for coronary artery disease, and they likewise found this attenuation of estimates. So this isn't to say for sure that that these effects are definitely through survivor bias, but they're suggestive of this. And I just want to highlight that these adjustments are made for a genetic liability to coronary artery disease and not necessarily development of coronary artery disease itself. What this means is that there could be residual survivor bias through this mechanism. And indeed, for any of these traits, and also ACE inhibitors that influence diseases earlier in life than dementia, that have a high survival rate, low survival rate, high mortality rate, this is obviously another mechanism of survivor bias. So there could be various paths of residual survivor bias here. Okay, so these potential caveats of interpretation and maybe limitations aside, I just wanted to very briefly touch upon some more recent work, which has attempted to identify potential novel pharmacological targets for Alzheimer's as opposed to exploring repurposing opportunities of approved medications. So in this paper by Su et al., the authors used combined blood and brain gene expression data for around 6,000 druggable genes. They tested this for association with Alzheimer's, and they found consistent evidence across both the blood and brain that these genes appear to be implicated in Alzheimer's. So for example, EPHX2 encodes soluble epoxide hydrolase. There's some evidence from mouse models of Alzheimer's that this appears to increase neuroinflammation. This is another very interesting paper where the authors had these relatively novel measures of proteins in the brain. So they had 400 measures of proteins in the brain. They constructed genetic instruments for these proteins and tested these for association with Alzheimer's. And using both Mendelian randomization and colocalization analysis, which Verena had discussed before, they found consistent evidence for 11 of these proteins in the brain being associated with Alzheimer's, many of them being novel, I believe. Okay, so I just wanted to wrap up by very briefly mentioning some possible future directions of relevance to drug target Mendelian randomization dementia. So beyond bulk brain RNA sequencing and protein data, as Verena mentioned just prior to this, increasingly there's a lot of really exciting brain single cell data becoming available, as you heard previously, and also brain region specific gene expression data available. Also in UK Biobank, there's new neuroimaging data and also exome sequencing data that's coming out. And I think going forward, integrating this data into a drug target Mendelian randomization framework will permit us to test increasingly more refined causal questions relating to both approved and potential novel drug targets at the molecular level. So I just wanted to conclude that aside from some of the caveats and limitations I mentioned before, I think drug target Mendelian randomization can still be an incredibly or potentially effective approach for both assessing the repurposing potential of approved drugs and identifying novel drug targets for dementia prevention. I think this will be facilitated by the increased availability of high-resolution omics data, in addition to the imaging and sequencing data that I just discussed. But I think careful consideration of some of the issues that I mentioned here, which are relatively unique to dementia, so nuances around the blood-brain barrier, survivor bias, is required in order for us to be confident in the robustness and interpretation of our findings. Thank you very much. I'm happy to take any questions.
Thanks, James. Questions? Yes. Yeah.
Great, thanks very much. Sorry, David Owen, Imperial. Thanks for the talk, which is very interesting. But I am really quite confused because so how do we? I mean, I take the point that taking a drug for 3 or 4 years for a particular time in your life is different from having a collection of genes which affects your blood pressure. But if we've learned earlier in the day that there's no genetic evidence that blood pressure is associated with Alzheimer's, but we also have no shortage of blood pressure randomized controlled trials, which is, you've shown in meta-analysis, showing that if you take these drugs, your risk of Alzheimer's is lowered. How do we?
Yeah, so I agree. There's a lot of what appears to be contradictory evidence. So I showed that there's evidence. I think the median age at baseline in the clinical trials. I mean, I can go back. So that was a meta-analysis of, I think, 10 or 12 clinical trials, which appeared to show that these antihypertensive, so they kind of grouped antihypertensive medications together. Obviously, they're given to individuals with hypertension. Yeah, I mean, they found evidence that this appeared to reduce risk of dementia. So when thinking about the data, I'm aware of contradictions between the conventional observational epi, where it appears that hypertension is associated with an increased risk of dementia. So some of the blood pressure data suggests a protective effect. And some of it, I think what Neil presented suggested possibly null effect. So, I mean, there's a few ways this can be interpreted. So one is that So one is that potentially survivor bias, as I mentioned before, could account for the illusion of a protective association. The genetic associations, even if they're measured in later life, could be picking up primarily associations at a different point in life in case there's some unique window where that's having an effect. Individuals with genetic liability to having higher systolic blood pressure are more likely to take, they're less likely to take antihypertensive medications. This could lead to an attenuation of certain associations. And it's also possible. Again, I don't know how plausible this is because I don't have expertise in antihypertensive medications. It's possible. So if medications have off-target effects, that could be driving effects in part that are not through blood pressure. I'm not saying this is very plausible, but yeah.
Sorry, those, we have so many randomized controlled trials of blood pressure lowering with a number of different types of drugs, which will all have very different off-target effects. So it is hard to believe.
So it kind of, I forgot the breakdown of the different clinical trials included in that meta-analysis. I don't know if a large proportion were testing perhaps a single medication.
Unlikely, I would have thought. I
would think so as well. I'm just trying to brainstorm, but possibility. There's quite a few possibilities here. My sense is that survivor bias is influencing a lot of dementia Mendelian randomization work that needs to be taken into account. And I think more agreement with the conventional observational analysis will be seen then. But yeah, that's just my sense.
But I think those trials were not done for dementia. That was not the primary outcome of the trial. It just happened that because there was interest later on, they reanalyzed the clinical trials data in relation to dementia. So I think it cannot be interpreted as strong as a...
But to be fair, I think the observational epi was also looking at risk. Like I think they're looking at the same outcome, which is incidence dementia. So
Jay. Pick that up in a second, but so I won't speak so long to lose that point. But great talk. Just a minor comment. We've got to be very, very careful about the ambiguation that often occurs between dementia and Alzheimer's disease.
Yeah.
When you're developing the genetic instrument for Alzheimer's disease, you're not developing a genetic instrument for dementia, which has multiple other causes.
Yeah.
But actually, the real thing was to have you think out loud and maybe think as a group a little bit more about how we can use the imaging data in studies like Biobank relatively more effectively, because the question is, I mean, we have a really remarkable opportunity to probe it or to probe multiple features and to look at genetic correlations of those, and through that to develop genetic instruments for the features. And those could be used more powerfully as diagnostic tools, specific diagnostic tools using MR. I mean, for the discovery of diagnoses in single or multiple diseases by looking at MR, so looking at MRI or other imaging trays that are causally linked to the disease outcome. Could you talk through how you were thinking of it? Because I think it's a really exciting idea.
Just propose would be something interesting to explore. I didn't have any very specific hypotheses, but what you just said there seems like something interesting that we could discuss further.
It's fantastic stuff. I mean, one thing perhaps to quickly check is that some ACE inhibitors cross the blood-brain barrier, some don't. So to see whether you see it, and you've got a lovely propensity matched population to do that with. And I mention this because out that that ARBs, so a different hitting a different part of the RAS pathway that cross the blood-brain barrier, but only those that cross the blood-brain barrier appear to lower the risk of Parkinson's by 50%. So there could actually be a different site of action, and that could explain the fact that this lack of the link between the vasculature and Alzheimer's. Actually, it might even have effect directly somewhere in the brain. Yeah,
that's a good. By my very basic understanding that ACE different types of ACE inhibitors have different abilities to pass through the blood-brain barrier. Actually, I agreed.
Any further questions? Well, if not, thank you very much, James. Thank you.